Hello everybody, good morning, afternoon, evening. My name's Declan O'Shea. I'm the product manager for isolators and containment systems here at ILC Dover. And thank you very much for attending our webinar, Controlled Environments, Safe Handling of Oxygen and Humidity Sensitive Products within Single-Use Containment Technology. So if you are familiar to the webinar series that we do, kind of present a bit of an insight into certain parts of the containment industry and containment applications that may be useful to yourselves as drug manufacturers or contract manufacturing or research and development groups in order to really understand what is possible within single-use isolator technology, and then also just within the general let's say scope of life sciences, to kind of give you a bit of an insight to see where you could take your new product or process in the future. So as always, kind of run down a little bit of a summary of where we're going to take this today. First of all, I'll start off with an overview of the company, just ILC Dover and its product offering in general. Then we're going to talk about creating the right environment. How would you decide what environment is needed for what process, and what do I really mean by environment? You'll hear me refer to this quite a lot as opposed to containment or isolators. I generally refer to what you're creating as an environment, and I'll explain that a little bit. Then we'll talk about our involvement as containment specialists, how our experience hones into being able to, dish out this guidance and knowledge, based on our previous experiences mainly, and just show you where that comes from. And then we'll look at process considerations. What do you need to do in order to maintain the environment that you're looking for? What kind of gases should you be using? What kind of air injection? What kind of room? Safety considerations, things like that. Then we'll go through 4 different operations, basically discussing, to what extent do you need to control the system with mechanical and operator controls in order to get what you want out of your process. And we start with a basic operation with manual gas flow, then sort of an elevated process using gas mixing and flow control. Then we'll look at something a bit more complex in around sort of purging and recycling. And then we'll look at kind of really unique ppm level applications and anaerobic environments using purge and catalyst control in order to get the environment that you need. And then we'll look at some of the more demanding processes. If you are combining low oxygen and humidity with something else, maybe a clean air environment, maybe high potency products, maybe you need to control your environment to a specific value rather than just provide a blanket. We'll talk about how that is possible. And then we'll just have a summary at the end, just saying, these sort of the proven benefits that the system can have to cater for the process that you're looking to use. So to quickly start with ILC Dover as a company, primarily we are a manufacturing and engineering company. We engineer solutions for our customers, requests and queries, problems into solutions, if you like. Started, sort of pre-World War II with manufacturing latex products. ILC stands for the International Latex Company. And then the Dover is because it's based in Dover in Delaware, or just outside Dover in Delaware in the US. But we've grown since sort of making army uniforms in World War II through to making spacesuits for the NASA missions. We still do that today. Through to making a lot of pharmaceutical products, making single-use bag systems for powder transfer, powder containment. Now moving into the biologic space with liquid bags and fluid transfer assemblies. And then we also make isolator systems, which I'm going to go over in a moment. We have presence all around the world, North America, Europe, and now a large presence growing in Asia. We've just hired just under 40 new people into Asia now, with much bigger reach out in that region, to see where our products can help out there. So, I was touching on the products that we have, primarily in life sciences. We work in the pharmaceutical and biopharmaceutical industry. Pharmaceuticals are a lot of powder transfer systems with flexible IBCs, powder bags for excipients and products. And then flexible containment systems, which is what we're going to talk about today. PPE, we have quite a broad range of personal protective equipment. And then in the biopharma side, we handle a lot of single-use powder applications, medias, buffers, things like that. Sterile manufacturing isolators, creating grade A, sterile airflow, and then liquid handling solutions in the form of 2D liquid bags. Now, what I'm going to talk about today will be a crossover between pharmaceutical and biopharmaceutical, I would say. We've seen low humidity and low oxygen being required in both of these spaces, depending on the application. And then some of those applications are what we're going to go over today. So to kind of kick things off, when I talk about an environment, what I'm saying is that generally, in order for your product to be handled inside of a place where you are going to not see any cross-contamination or any adverse effect while handling the product, and then the same where you're not going to induce any hazardous effects to your operator, you are generally creating an environment that is suitable for both sides of the process. So yes, you can call it containment, you can call it an isolator, but realistically, how the air flows in and around the system is what creates that suitable environment and that working space for the product and operator. So going through sort of some of the different examples, if we're talking about cell banking, a lot of the time you're looking at low oxygen anaerobic environments, for which we talk about nitrogen gas saturation, which is the topic of today. Then move to something like ADC compounding, where you're looking at highly potent toxic materials. You'd be looking at a high containment system using a closed barrier, negative pressure airflow. Or maybe you're looking at fill finish, where we look at a sterile environment with a low particle count, low bioburden, in order to maintain true sterility in that process. And then maybe you're looking at dry powder inhalation. You need a low humidity in order to prevent any moisture gain there. And then we'd look at sort of compressed dry air saturation, which again, is something we're going to talk about today. And then talk about the difference between these. But what I'm trying to get across essentially is that you can pretty much utilize most systems to operate in, but if you don't create the right environment for your product, your process is not going to work correctly. So, I'd just be cautious when you're looking towards handling specific products or processes that you consider what environment you're going to be using. So, how can we speak towards what we're speaking to today? So, my experience comes across namely from being in the sales team, sales engineer, then a sales manager, moved to be a product manager. And really, it's the fact that as containment specialists, we are approached by customers with a broad range of applications across all industry sectors, not just life sciences. So approached by the nuclear industry, the atomic industry, for any sort of containment application where we find that there is a requirement for containing hazardous materials or protecting a product inside. In the nuclear industry, for instance, there are a lot of anaerobic glove boxes that are used, and we see requests coming in from that side. And I'd say, as a company, we see, maybe 3,000, 4,000, 5,000 different opportunities and inquiries every year. Yes, we don't necessarily convert all of those, but we do gain experience every time we approach a new opportunity as far as saying, how can we solve this? And then what we do is we kind of tie all those combinations together to create a single portfolio of new product innovations, which can lend to all the markets by using the experience of different sectors. Because ultimately, in the end, containment is required all over, not just in farm and biofarm. So we try and lend those experiences the same to cater for those sectors. So in our case today, we've taken our experience from those sectors, from those past inquiries and installations, in order to really maintain sensitive environments, namely talking about products, because that's generally what we're going to be protecting under low humidity and low oxygen, is we're looking at protecting the product itself and ensuring that it can operate within the process that you're looking at within the environment that we're going to provide. So process considerations. I mean, you're going to ask yourself at the beginning of your process, or before you even conceive how you're going to do this, what is the best way about doing so? I know, for one, that I'm going to need a low oxygen environment. We need to work under sort of 1 or 2% oxygen in order for this product not to oxidize and change form. If you do, then that product will be lost, the batch is lost, and then that's money and time down the drain. So you need to make sure you observe those those conditions inside. So what is the best way to go? So realistically, when you're looking at gas usage, a lot of people will ask, what gas do you recommend? The most common is probably nitrogen. Second comes argon. But really, any inert gas which can offset or a heavier gas than oxygen to offset the oxygen content within the closed chamber can really be used for any of these applications. We just generally find that nitrogen is a lot easier to generate and come by, be it in bottles or with a nitrogen generator on site. Generally, a lot of larger facilities will have those. That's generally what you're going to be needing. Sometimes in extremely low conditions where you're looking at maybe ppm or sub 1% oxygen levels, you'll be looking at pure nitrogen or pure inert gases in order to really obtain the levels that you're looking for. I know with certain anaerobic cell banking practices, some of the levels you're looking at is 100 ppm, 10 ppm in some very rare occasions. And for that, you would require, an extra pure form of nitrogen in order to ensure that the oxygen content has been scrubbed down. And then lastly, a lot of people ask about low humidity environments. Do we need to have a nitrogen source for that? Not necessarily. If you have a source of compressed dry air inside, all you're looking at really is taking the moisture out of the air rather than replacing the air content inside. So if you can have a supply of compressed dry air, which is generally a lot more readily available, it can be a more cost-effective way of reducing the humidity inside of your containment system as opposed to nitrogen. And usually the flow can happen in exactly the same way. You'll find a lot of systems can switch very easily between a nitrogen supply and a compressed air supply, depending on the the effects that you're looking to obtain inside. Now, this is not usually something that we speak about in these sort of talks because it comes, I would say, naturally to people when handling potent products. But some people sometimes don't necessarily think of safety considerations when it comes to handling inert gas. So compressed air, that's not going to do anything to you really. However, it might give you a dry throat if you're in a room for a long time without it, become very uncomfortable and you'd probably leave. But ultimately, in the end, it's probably not going to kill you before you realize. However, if you're looking at using inert gases, so say nitrogen being the most common, if that is to fill up a room, a lot of the times, the operator is not going to know before they fall asleep. And when they fall asleep, a lot of the times it can unfortunately be permanent. So you need to make sure that you are working with an airtight piece of equipment in either a well-ventilated space or in a room that has enough sensors and enough means to exhaust the air from the room or replace the air in the room in case of a leak, that you are taking those safety considerations into effect. I've seen it in time where people will fill sort of 2 or 3 cubic meters of nitrogen in a room, the size of a janitor's closet, and then are wondering what the safety concerns are with doing that if they don't have any measuring or any way of exhausting that air from there. So please do consider your safety concerns when handling the gas. And then more of a practical concern, things like gas bottles can become, I just put here, mini rockets. Essentially, if they're not handled properly, if they're dropped or ignited in any way, then there can be grave concerns with those things flying around the room. So please ensure that they're in a dedicated rack or tied back to a wall, or consider your safety concerns when handling those. Now, what we're going to get onto now is essentially the 4 different types of systems, based upon the levels of control that you require. So when you're looking at obtaining environments inside of an isolator, not all of the time you're going to require such strict, considerations around your process. Sometimes you may not require 1% relative humidity or 1% oxygen or even 10%. You might just want to provide a blanket for a very basic experiment where you know that you need below 10% oxygen, maybe 20% content inside. So why should you have to purchase a system that has such comprehensive gassing methods on, when a lot of the time it can be done manually? Yes, okay, a manual process can be unpredictable and can be more labor incumbent. However, if you're working on a research and development process, or in a place where you may not have the capital to invest in this equipment, a simple containment bubble like the one that you see in front of you here, with a simple nitrogen controlled nitrogen inlet at the rear, and then exhausting a ventilated sleeve through maybe a biosafety cabinet or a fume hood, something with good extract out of the room. You can purge down a system like this to sort of under 10% in a relatively quick time. I think when we did tests on this unit, we got it down in about 12 to 15 minutes in order to get around 10% oxygen content inside. And then you could do exactly the same when you're looking at it with compressed air for low humidity. So, as it's stating here, you're going to load all your materials inside. It's a one pass-through operation, so make sure that you've got your checklist done and everything's inside before you work. Inflate the containment system with nitrogen. Have an O2 meter inside so you can read the oxygen content as you go. Make sure that you're venting the nitrogen off the top of the isolator through the sleeve provided, in order so it doesn't blow up, exponentially, and then possibly rupture the system. It would take a lot, but it is possible. And then just balance that gas supply manually with a sort of trickle feed in order to maintain the oxygen content that you require. We, of course, can show you exactly how to do this. We've got full manuals on how to operate basic systems like this, and for some people, this is pretty much all that you would require to create that very basic environment that you want at a very low relative cost. Perhaps you're actually looking for something a little bit more. Perhaps you're looking for a more controlled environment, maybe some more accurate readings, something that you don't have to manually control. Then you consider sort of the next step. You're not going to go the whole hog. You don't want a fully controlled PLC system. You want some kind of halfway system that will do for your process, that you're going to have sort of repeatability out. So when you look towards a system, this is just an example of one we've provided before, where you have a 6-sided contained system, and then you have a gassing tower on the side. Now, this gassing tower would allow you to set a trickle feed of nitrogen or compressed air into the system in order to saturate the environment. And then the exhaust fan draws a pressure feedback from the main chamber to exhaust the air out at a steady rate that balances with the air coming in. So essentially, through some calculations that, of course, we will provide and set up for you, we could maintain a negative pressure based on the fan speed with the injection of nitrogen in order to keep a constantly saturated environment while you're working inside. Now, these systems here have been proven down to sort of 1% oxygen content inside, and a relatively controlled state. So if you look at the tower to the right-hand side there's a little blue control panel on top. That's the oxygen sensor that feeds back from inside the isolator. And then the larger white panel will show you the air pressure inside, or the differential pressure, as well as the air speed in order to maintain that environment. And then with a system like this, if you want to transfer materials in and out easily, you can use something like a rapid transfer port in order to not disturb the oxygen levels inside. The one thing that you would have to consider, of course, is that if you are transferring materials in through a port like that, is that that canister itself with the materials inside may contain room levels of oxygen, or wherever the previous transfer has gone through, or if it's been sat for a while, it might not contain sort of an airtight anaerobic environment, so that will spike the content inside the isolator briefly when you're making the transfer. So it's not a true anaerobic process, but realistically for a development operation or sort of small quantities of product, it's a nice little halfway between the very manual labor intensive process and then maybe something that would cost 2 to 3 times this in a more comprehensive controlled process. So if you want to take things one step further, which of course a lot of people would do, if you're running a process that requires a lot more control and a lot more, let's say, reassurance in regards to the levels that you have, with a bit more flexibility, you could opt for a more comprehensive system. So you could look towards having an environment which was made of 2 chambers. Perhaps you wanted to load materials and pass them through, guaranteeing that you have the same environment in your antechamber as well as your main chamber. But then you might also want to record this data. You might want to have feedback from the sensors. Maybe you're running under a 21 CFR Part 11 compliant, data integrity scheme at your facility, and you need to record that data. Something like this would provide all of that for you, as well as allowing you to change the set points in order to say, okay, for this particular process, we're going to require a 5% oxygen content inside. We maybe don't need to use that much nitrogen for that longer time. So let's create that set point to there. And then maybe you're running a different process the next day, with a different product, single-use film. You can make the changeovers very easily and efficiently. And then you change your set point down to maybe 1% oxygen or relative humidity content for the next process. So a gassing unit like this with a versatile PLC will really allow you to change that process up in order to provide something that's a little more versatile than the previous 2 units that you've seen there. With, of course, some added safety features, where you've got proximity switches on the airlock to shut down the gas supply if anything untoward is to happen there. And then you've also got sort of in-chamber and in-room alarms that would sound if the atmospheres or environment, were out of the normal ranges that you were going to be looking at. So you don't necessarily have to go for such a comprehensive system like this if your process does not truly require it. However, the options are still there if you require something that's maybe a little more mechanical control. Then the final one that we're going to talk about as far as examples for today is probably the most unique and certainly the newest for us, is that we, in cooperation with a partner of ours, have developed a system where we've created a single-use isolator chamber, which is a 6-sided structure that can be fully disposed without any sort of required cleaning validation. You'd simply place up a new film and start on your next process. But we worked with a customer that required ppm levels of oxygen inside their process. Now, this was for anaerobic cell banking that I mentioned before with a customer in North America. And what they required was a 10 ppm environment to be maintained for a number of days, a number of weeks actually. It was about 16 days in total that they needed to maintain this environment. So we knew, what do we have to look at here in order to ensure that we maintain that level of environment for such a long time. Bear in mind that when we're talking ppm, this is parts per million of oxygen within a certain volume of air. It's unfathomable to consider these sorts of levels under normal circumstances. Hence why we sought to cooperate with a specialist that knows how to handle these environments. So what we did from an isolator perspective is we looked at all the possible leak pathways that could be developed when you have a system like this. So we utilize radio frequency for gas tight and airtight welding, as well as YKK gas tight zippers. And then the interfacing with the stainless steel section that I'm going to talk about in a second is made of a gripping system that we use for pressure decay functions. And we thought, you know, what we have to do is engineer out as much of the leak pathways as possible to create a single airtight system. So this means removing any flexible airlocks, removing any additional sleeves or entry and exit penetrations that don't need to be there, and really ensuring that everything that we manufacture here is as robust as possible. Now, what we did for the ppm levels of oxygen is that we figured out that you can probably get with purging and recycling the air and then back purging the isolator again, you could probably get down to around 100 ppm over the course of an hour, maybe an hour and a half, by constantly doing that cycle. However, the usage of nitrogen in that situation was extreme. And then we also found that really you could not get below 100 ppm effectively with using that method. So what we did in the end is we actually consulted a company that creates anaerobic glove boxes for the nuclear industry, and they introduced us to a form of doing it with palladium catalysts, where they will run a purge cycle down to a few hundred parts per million of oxygen, and then they'll run the rest of the air in the chamber once purged through the catalysts in order to oxidize the rest of the oxygen that's inside, leaving just pure nitrogen, leaving a pure nitrogen environment. Once that purge cycle's been done, you just have to work out what the natural leak rate is of the unit in order to provide, you know, a steady trickle of air coming back through the isolator from the nitrogen feed. So using this combination of a purge down system plus air scrubbing, you can break down from 100 ppm down to, you know, sub 10 ppm. We actually did an experiment which lasted about 7 or 8 weeks, where we managed to hold about 5 ppm for that duration inside a single-use flexible system, meaning that we knew that everything was airtight, everything was integral, and all it required was a very low level of top-up to maintain the oxygen, maintain the low oxygen levels inside. And realistically, these systems can be made for any manufacturing process where you have a 6-sided environment that you can place process equipment inside or process or product, and then work under these conditions. Now, one thing that we have to advise with this is that, you know, you really need to focus on entry and exit from the system. You know, are you going to be loading other materials throughout your process? You're going to need to maintain this environment for a number of days or weeks. Consider, you know, how often am I replacing something inside? What is going to happen when I introduce new materials? When we're working down at ppm levels, you have to consider that quite literally every single particle of air matters. So when you're introducing new materials, you've got to consider there may be air bubbles inside the packaging. There may be, you know, maybe dead spots within the containers that you're placing in, and anything that introduces several hundred particles of oxygen inside that's come from another process will alter the whole environment inside the isolator. Yes, this can be overcome, but do consider to keep these processes down to a minimum. Hence why you see on the back plate here, you'll see that you have a larger square surface for inbound loading of the original materials, and then a smaller circular port, which requires much less purging time to inbound ancillary material. Now, going on to something that might be a little bit more demanding. You know, so I'm speaking relative terms with the previous examples towards a single process type. So I'm saying low oxygen, low humidity, using nitrogen or compressed dry air only to create that environment. But what happens if you require multiple environments? Some processes are not as simple as low oxygen or low humidity only. You might need different operations going on site, working with different products. So how are we to combine the different types of systems in order to create the right solution for you? So I've come up with a few examples that we have here, and these are all examples that we've catered towards before. And as I said right at the beginning of the presentation, as containment specialists, we have a broad range of knowledge across many different industries that allow us to combine these together to create the correct environments and combine our previous solutions that we know have worked, independently, to bring them together to say, okay, we made this work in this way, we made this one work in this way, so how can we make them work together? So if you think about low oxygen in a sterile environment, so what you can do is you can link up something like a VHP biodecontamination system with your closed isolator. You already need to run pressure decay cycles. You're already going to have a good amount of airflow. So why not allow peroxide purging inside your unit, create that kill cycle, get your log6 sterilization to work with a sterile product inside. And then the aeration can come naturally in the unit because we're already looking at higher airflows when we're working with these nitrogen-based systems anyway. So that is certainly a possibility, that if you need something that's a bit more sterile, you can also work with low oxygen. So what happens if you need sort of low humidity and then a higher airflow? So you need maybe a bioburden free or as low bioburden or particle count as you would like. Maybe we're looking at something like a compressed air fed high flow isolator. So using sort of powerful fan units and a supply of compressed air, we could generate an ISO 5 particle count using 20 to 30 air changes an hour inside of your chamber. We find if we can generate that sort of airflow, that we can bring the particle count down inside to ISO 5 standards, not a problem. And if we're using compressed dry air to do that, all we're doing is replacing that regular room air with a more dry source, and then naturally the humidity inside of your unit will come down. The only caveat around this one is you need to make sure that you have a powerful enough compressed dry air source that can feed the amount of air changes that are going through the isolator. Most systems can, but just bear in mind if you're in an older laboratory or an older clean room that doesn't necessarily have the same supply, to make sure that you check that beforehand. High containment and low oxygen is quite popular at the moment. I talked about antibody drug conjugates, or ADCs, at the beginning of this presentation. And essentially, yes, we found sort of about half of the ADC applications we've worked with require a low oxygen content for a sensitive product. And in that case, a lot of the examples I've shown with a 6-sided fully disposable system coupled up with a nitrogen purging tower can really accommodate that process. So just because you're thinking this product has high potency risks around it, doesn't mean that we can't integrate other features into the unit and still make it an airtight low oxygen system. And then the final example I placed here is around humidity control. I would say this isn't the most popular, but it certainly is something that we've seen as a request from customers before, is that you have a certain humidity in the room. So you have a background humidity, and then you have one process that needs to run at humidity A, and then you have a second process that needs to run at humidity B. But you really don't want to dedicate one room to one product or process, especially if you're in contract manufacturing and you don't have any cross-contamination concerns. But you would like to maintain 2 different environments inside the same clean room. Now, what we find is if you have a gas mixing system which effectively mixes room air with compressed air to maintain a certain humidity inside with some sort of humidity feedback loop, is that we can keep one system at, say, between 10 and 15% humidity, and we could keep another system at between 5 and 10% humidity, therefore allowing you to run two different processes inside the same room, but utilizing two different systems, essentially saving yourselves on footprint. So just because you may think that your process is a lot more complicated or has many different steps or considerations, it doesn't mean that there isn't a solution out there for it. You'll find that a lot of containment providers are approached by many different customers to contain many different products and processes, and often they can add two different processes together to come up with a solution that you're looking for. So the one final thing here, just kind of take homes for today's talk is, the choices that you put into it and safety really do matter. Make sure before you want to run this process or even purchase any equipment associated, is just make sure you're choosing the right sort of input. Are you using nitrogen where compressed dry air could be used? Are you trying to use nitrogen when you might need a more pure source of it in order to obtain lower levels? What sort of impact is that going to have on the room and the facility and the operators around it? Sometimes people can jump feet first into these applications without truly considering all the background that might be required. So, take away what I've given you today to say, let's look into these other things. Am I suitable within the environment that I'm working in order to handle this product or process? If yes, let's proceed on with the equipment suppliers and say, we can run down this process. I know that my room and operators can cope with it. Much to the same fact, always run a risk assessment on this process. You're handling, something that is potentially hazardous to the operator here. Yes, you might not be handling a potent product and that might not be the immediate risk. However, inert gases are a risk, and I would say make sure you're well equipped for handling them within the space that you're looking at using. And then finally, choose a system that is applicable to your process. There is no point in spending hundreds of thousands of dollars on a system that you could have a lot more manual or semi-automated intervention in. Perhaps you're in a process that is not, fully thought out. You're still in early stages of development, and you don't want to make those kind of commitments. Just know that there are solutions out there that don't need to cost an absolute fortune. Investigate what your conditions are going to be, and then choose the right system for those conditions. Have a think about how much control you want to apply to the process. How much do you want to control the nitrogen level inside? How much do you need to control the other background environments? What kind of mechanical controls versus operator controls do you have to weigh up in order to make sure that the process works correctly to what you can do? Also consider scaling up. Some of these systems can be purchased in two halves. You could purchase the isolator itself with a different gassing system or airflow system, then maybe upgrade that airflow system if you require further control further down the process, or with a different product or process you're choosing to implement. And then make sure you break down the limitations. There often are going to be limitations with handling this, and then always look towards mechanical controls first before you look towards operator controls. It's much more safety assured and process assured if you can allow a mechanical control to take over, bridge that gap of risk, and then ensure that the operator is working safely, and then they can cover anything that mechanical controls feasibly can't. So that's it for today. I understand it's a little bit shorter than the usual presentations that I have. However, I hope everybody has learned something from the handling of this today. And then just know that there are lots of possibilities out there for creating anaerobic environments inside maybe a more cost-effective system and maybe a disposable system if you're worried about cross-contamination or product contact or cleaning validation of any of the systems that you have. If anybody has any questions or queries, by all means, you can place them in the chat briefly now, or if you want to email or call me directly, that's absolutely fine, and I would be happy to talk about anything we've talked about today, or expanding this into different areas, or if you have a particular application in mind, we can talk over that, not a problem at all. So if there's nothing else, thank you everybody for your attention today, and enjoy the rest of your week. Thank you.